Dubla ko na eh. Hello, good PM. So in this video, we are going to discuss the rearing techniques of for mites. So mites are reared for a wide range of primary agricultural, public health research, aesthetic, and other purposes. Mites rearing may be partial, egg to larvae, or nymphs and adult. There are a number of methods for rearing of free-living mites that are developed. They are cultured in various types of glass or periglass cells. So, yung mites ay inaalagaan for many purposes, like for example in agricultural purposes. Yung mga farmers and yung entomologists, scientists is inaalagaan nila yung mites for experimental para sa mga scientists and entomologists, and sa mga farmers is the rare mites for using them as a biocontrol of spiders, two-spotted spider mites, or any parasitic mites that destroys their plants. So mites, so yung mites are reared may in partial. Pwede inalagaan nila yung mites pwede sa eggs to larvae or nymphs and adult. At dahil nga sa maraming purposes ng rearing mites, marami na rin yung methods of rearing for free living mites na na-develop. So, yung mites ay nakukultured and various types of glass or periglass cells, and pwede rin sa plastic containers. So, different techniques and or methods of rearing mites. Number one is the mass rearing techniques of mites. The rearing container consists of two inner and outer plastic boxes. The inner box contains rice husk and is placed inside the outer box, which contains a small amount of water. The primate Tyrophagus protescentiac was also reared in similar way using rice bran and provided to G. aculicer once a week. The rearing condition within the inner box were maintained at 25 positive negative 1 degrees Celsius and 95 positive negative at 5% relative humidity. So, yung rearing container ng mass rearing techniques of mites is we have two double box sa una doon nila yung nilalagay yung rice husk, tapos yung rice husk yung at saka yung small amount of water, tapos yung prey, tapos sa isang room naman is yung G. aculifer, tapos yung rice bran. So dito sa technique na ito is that they maintain the condition of the inner box, and they maintain the relative humidity na meron lang yung 65 yung specific na level nun. Tapos in this double box system, the G. aculifer was rapidly grown and overpopulated within 2 weeks only, while the another predatory mite is like the Stratiao lilaps or is schimotos is 4 weeks period siyang na-overpopulated. Tapos ito yung itsura ng container ng mass rearing techniques of mites. So made in plastic siya, yung not in glass. So the number 2 is the modified leaf arena technique. Is a new procedure for rearing mites in the family Phytocidae or Tetranychidae. The rearing units provide air exchange and stable level of humidity, specifically 75 to 85 relative humidity, and the microenvironment at 1 centimeter height only. At 26 degrees Celsius or less, mature citrus leaves were maintained unit of in the unit up to 8 weeks. Tapos, pollen of the ice plant or the Malifera croquet was usable for 10 to 15 days as food for uh, Eucelios mesimbranus at 18 to 30 degrees Celsius. So, in this method or techniques, the level of humidity is maintained also, and and tapos yung infested leaf is maintained also in the unit up to 8 weeks. So the pollen of the ice plant in the Melafora is ginagamit yun as foods of Euceus mesimbranus na naaalagaan in this technique. So the method requires low maintenance, is simply relatively inexpensive, and allow monitoring of mites behavior or development through the clear plastic. So ito yung itsura nun. So meron siyang sa letra A, yung itong is made on plastic yung container niya, tapos glass naman sa letter C. So this is the leaves of yung letter B is the leaves of a coffee which is infested by mites. So the number 3 is tertiary leaf cage technique, and then number 4 techniques or method is the leaf disc method. And this method is that the prey remain in good condition and in some cases may reproduce. Tapos, however, not all plant species are suitable because leaves may not last once excised, and leaf supporting media cotton wool, cellulocotton, and polyurethane that do not always present escape of prey and predator mites. So, as leaf supporting of media, they use cotton wool, cellulocotton, and polyurethane. Pero yung ito is it does not prevent the escaping of prey and the predator mites. Tapos syempre in this leaf method also is the prey and the predator is maintained, is reared with a good condition also for them to reproduce. And leaf this method also needs cotton swabs to be moistened periodically to avoid drying of leaf this. So yun, kailangan ng method na ito yung cotton swabs para ma-moisten siya. Tapos kung minsan ginagamit nila yung moisten filter paper as a alternative of cotton, or alternative it's they use filter paper, moisten filter paper as instead of cotton. And so ito yung halimbawa ng rearing container of that is used in leaf disc method. So ito yung nung example of the container of the leaf disc method. So the next method is the Telemethod. This was developed by Glenn Scriven and James McMurtry on UC Riverside. So the materials in these methods were a shallow pan that is filled with 1/2 inch of water, 1 inch moisture foam cut to fit pan, squeeze in water until soaked, black plastic or vinyl tile cut to foam. Leave a narrow margin of foam exposed, tissue cut to fit around the edge of the black plastic, drape it over the edge of the foam and touch the water. So the spider mats were placed in infested leaves on the black plastic and a leaf that is infested also by the predatory mites. So dalawa yung mites, klase ng mites na nilalagay dito. The spider mite, and the predatory mites. So in this method, always maintain correct water level. If only a few live spider mites remain, add more infested leaves and keep them in the center of the tile or plastic to prevent moldy leaves. So this is an example of mites that are reared using the techniques that I discussed a while ago. So number one is the rearing technique of mite Trenicles or TC. So the samples of eggplant leaves infested by Tetranicus or Tinaceae all samples are placed on bags and transferred immediately into the laboratory. The mass culture was initiated by transferred individuals of females and males using a camel's hair brush placed in petri dishes 10 cm diameter, which provided with untreated fresh leaves this of mulberry, about 2.4 cm diameter, placed on a pad of cotton wool fully saturated with water as a source of moisture and to prevent the mite escaping. The newly laid eggs and hatched larvae were placed on fresh leaf discs in prepared petri dishes as mentioned above. The old leaf discs were removed wherever it was necessary. All colonies were kept in an incubator at 27 positive negative 2 degrees Celsius and 65 positive negative of 5% relative humidity. So this is the uh, Tetranicos orticeae, and next example is the rearing techniques of predaceous mite, Phytociolus microphyllus. So yun yung picture niya. So the predaceous mite was reared on the kidney bean leaves, highly infested with T. orticeae, a spray that putted in large metal box 35 by 25 by 5 cm pieces of sponge 2 cm thick were put in the middles of each box. So potted filter paper on a sponge and leaving a piece provided every day as food source. The box were kept in an incubator at 27 positive negative 2 degrees Celsius and 65 positive negative 5% relative humidity. So the water was added daily to maintain suitable moisture for the predatory rearing. So these are my reference, and yes, rearing techniques for mites report by Judelyn Matias, Visa 3. Thank you.